Well, while we're getting set up, I want to say the most important thing of the talk, and that is thanks, Larry, for the development and the collaboration with the scientific community for Raman optical activity and VCD. And Rena, thank you as well. And it's a great pleasure to be able to offer congratulations and to talk about one of our favorite subjects and be given the opportunity to talk about biological structure and function and the use of vibrational spectroscopy in order to study that. Now, if I could have some photons with the laser, look at that, then I could tell you about some recent work in the UV spectral region to excite the Raman spectra of vibrational modes of chromophoric molecules, or molecular segments in the case of proteins. And this is a story of looking at molecular segments in order to interrogate their structure and function and dynamics. And this kind of spectroscopy, UV resonance Raman, gives rise to very high sensitivity because of the resonance enhancement effect and very high selectivity. And it allows you to focus into important regions and dynamics. It also avoids fluorescence interference with the Raman spectra. Now, this is not obvious at first that this would occur. It happens because it turns out that there's essentially no relaxed fluorescence for any molecules below 260 nanometers in the condensed phase. And that turns out to be the case because there are no good quantum states of molecules below 260 nanometers for their first singlet state where they would normally fluoresce. Essentially, all small molecule chromophores that have their first S1 state below 260 nanometers relax non-radiatively. We've never found interference in any deep UV Raman measurement from fluorescence. So this is very nice, and this allows us to monitor ultra-low concentrations of analytes. We have other applications such as standoff detection of evil molecules and biostuff. This enables excited state molecular structural investigations. At one point, we were doing CVD diamond studies in the plasma. The black body emission was not an interferent if you're exciting in the deep UV. And the subject here is examining protein structure and dynamics. And it turns out that you can get really incisive insight into protein and peptide folding. You can get a very deep look at alpha helix melting, for example. UV resonance Raman, or 1 to 2 minute to 5 minute fast measurements of solution spectra that allow you to definitively determine conformation about peptide bonds as a function of solution conditions. And what I'm going to tell you about is some of the first experimental determinations of the Gibbs free energy landscape along an important folding coordinate. And this is the first example of the experimental determination of a free energy landscape. And this has now constrained the calculation people in what they can imagine. So to remind you that Raman is an inelastic light scattering phenomenon, we excite our molecules with a large number of photons. We collect the Raman scattered light and interrogate that light for intensity and frequency and polarization, and that's what gives us molecular information. When we excite molecules, what we do is we interact with the electron cloud and we displace charge, and we displace charge in proportion to the electric field. And so when we excite with an oscillating electric field at some frequency, we create a dipole at that frequency. That dipole radiates light. If it radiates elastically like Rayleigh scattering, then the light is scattered at the same frequency, and our job is to get rid of it so we can look at the more interesting Raman scattered light. And that Raman scattered light is the result of the coupling of molecular vibrations with the electron cloud. And so you have a beat frequency, you have the Raman scattering, and that's a much weaker process. However, we can impact the efficiency of Raman scattering by choosing an excitation wavelength which is in resonance. And so by choosing an excitation which is in resonance, we can excite particular electrons in particular orbitals in particular regions of a molecule. And so you have the selectivity of forcing those dynamics. You also, because you're exciting in resonance, cause a larger dipole moment. And so you increase the efficiency of the Raman scattering process. And then what you can get is a large enhancement of the Raman scattering. And you get the Raman scattering of particular vibrations that couple to the electronic transition that you're exciting. And so this gives you very high efficiency and selectivity. And you can see that selectivity in this cartoon here, where we show the absorption spectrum of the protein myoglobin. And in the case of myoglobin, you have a heme, and that heme stores oxygen. And if you look at the absorption spectrum, in the visible, you see the heme absorption bands. Whereas as you go further in the UV, you see the absorption bands of the aromatic amino acids, and then the very steep absorption incline of the pi-to-pi star transitions of the peptide bond. And as we change our excitation wavelengths, we get different resonance Raman spectra. So in the case of exciting in the Serre band, in the near UV at 415 nanometers, we get a resonance Raman spectra dominated by the heme in-plane ring modes. And we can interrogate those modes to look at the conformation of the heme in its pocket. We can excite in other charge transfer bands to see ligands bound to the heme. If we get bored with that and want to ask questions more about the proteins, now we can do resonance Raman excitation, for example, in the shoulder here, in the absorption at 229 nanometers, which is in resonance with the aromatic amino acids that are sprinkled around the protein. And so we get different spectra and can ask different questions and talk about conformation and hydrogen bonding, for example. And now if you get even more bored, you can excite further in the UV in the rising pi-to-pi star transitions, and now you're measuring a UV resonance Raman spectra dominated by the peptide bond pi-to-pi star transitions. And what you're seeing are vibrational modes of the peptide bond, and those vibrational modes carry information on the conformation of those peptide bonds on the hydrogen bonding, and you can get a lot of information. The vibrational modes you see are vibrational modes of the secondary amides of the peptide bond, and you simultaneously see the amide 1 vibration, the amide 2 vibration, the amide 3 vibration, and there's a very nice story about why these modes are enhanced as you excite within, for our case, will be mainly the NV1 transition of the secondary amide pi-to-pi star transitions. The spectra that you measure, for example, for the PP2 conformation, the alpha helix, and the beta sheet, are quite different, and I show spectra in regions we understand, and I hide spectra we don't. But if you take a look at the parts of the spectra we understand, you see the amide 1, the amide 2, the amide 3 amide bands, and then you see another important band, which is the NC alpha H bending. These 4 bands give you a huge amount of information, as you'll see on the conformation. You'll also see that the frequencies and relative intensities are quite different between the different conformations. And so you can use these spectra, these basis spectra, to determine conformation. And in an early study of a library of 13 proteins whose X-ray structure had been determined, and thus whose conformations determined, you can take these basis spectra and fit them to the protein spectra, and from that fitting, what you can determine is the composition. So these are less than 5-minute spectra which you've done that, and it gives you composition with very high accuracy. And you can see that this identification of the weighting of the basis spectra is very good, because the residuals are also shown in the spectra. And you see, except for cytochrome C, there's very good agreement and very few little residuals. So this is a very powerful conformational method. Comparing it to the other fast spectral method, CD, there's a huge amount of additional information here that you don't have with CD. Be happy to talk about that in detail. Okay, so these spectra show that the different conformations have different Raman spectra. And what is the origin of the differences? And the origin of the differences is that the different secondary structures have different dihedral angles which depend on the conformation. These are the well-known Ramachandran psi and phi angles, and these define the secondary structure. And so if you take a look at the conformations that are possible for the peptide bond that's shown in a Ramachandran plot, where the psi and phi angles are displayed, and this is sort of like a contour plot of what's possible. And so the extended structures have psi values close to 180, the alpha helix minus 160, and a long time ago, Dick Lord drew this psi circle where he noted this correlation of the amide 3 frequency with the conformation, and pointed out that this might be a technique that's quite useful for determining conformation. Well, this turns out to have been exactly right. And the reason why there's such a strong conformational dependence can be seen from these two figures. The peptide bond has motion involving CN stretching and NH bending and C alpha H bending. And it turns out that in the NH bending, you find that it's coupled with C alpha H bending when the C alpha H and NH bond lie on the same side of the peptide, and there's intimate coupling. On the other hand, and that occurs in an extended conformation, like a beta sheet, for example. On the other hand, when you're in an alpha helix conformation, the C alpha H bond and the NH bond are trans to one another, and there is no coupling. And so the degree of coupling depends on the Ramachandran psi angle. And so what you have in theoretical calculations shown here is sort of a sinusoidal dependence of the frequency of a particular vibration. We call that the amide 3-3 vibration that we studied in detail with the help of Max Deem and some of his isotopic derivatives. And so we examined our theoretical calculation and a whole series of different crystals of small peptides with different psi angles to characterize this dependence. And what we found is a family of curves which has this sinusoidal dependence which only differ in the frequencies due to hydrogen bonding. And you can combine all of this information to come out with an equation which relates the amide 3-3 frequency to only 3 parameters. And that's the Ramachandran psi angle, the hydrogen bonding, and the temperature. So if you know the temperature, and if you know the hydrogen bonding, then you know the psi angle once you measure the amide 3-3 frequency. And this is a remarkable insight into structure and dynamics. And here, for example, are a series of spectra of polyglutamic acid, polyl-lysine, XAO. This is a synthetic peptide made by Kallenbach, I'm going to tell you about in a minute. Here is the spectrum of alanine 5, from which we subtracted alanine 3 to calculate the spectrum in the middle of this peptide. And this is a mainly polyalanine peptide. And what you see for XAO, which Kallenbach showed definitively by NMR, was a polyproline 2 conformation, is a spectrum that looks like this, which is identical to the center peptide of a small polyalanyl peptide, and is the same as that of AP, a 21 residue polyalanine peptide. Single conformation, average conformation, not random coil, but polyproline 2. This is quite different from the spectrum of polyl-lysine and polyglutamic acid. Okay. Well, we have a correlation of amide 3 frequency and psi angle, and so we have spectra that are inhomogeneously broadened. They have extra conformation, but yet we know how to correlate those. We also have measured for single crystals what the homogeneous line width of this vibrational mode is. So we can convolute the homogeneous line width out to calculate a psi angle distribution for the peptide bonds of our sample. And what we discover for polyl-lysine and polyglutamic acid is our band is lumpy and dumpy because it's got more than one conformation. In fact, it's got 2 conformations at 2 different psi angles. And one of those psi angles is polyproline 2. And the other psi angle, well, we thought we discovered a new conformation. It's called a 2.51 helix. And so we were very excited, and then remembered Sam Krim had theoretically predicted a conformation for charged side chains where they were splayed out due to the electrostatic interactions. And so this is another extended conformation which looks like this and has a psi angle of 170 degrees. So what we discover is that these charged side chains are in a conformational equilibrium between PB2 and 2.51 helix. Now that's a nice thing to know from the Raman spectra. You've got these 2 conformations in equilibrium. Actually, the amount of each tells you what the relative energy is. And if you take a look now at this conformational distribution in psi, you find out that it can be easily modeled in terms of 2 Gaussians, which is exactly what you would expect if you had a system defined by a torsional potential associated with the psi difference from the equilibrium state. And so you should have a Gaussian-like distribution, and which is exactly what you see. Now, if you can determine how much of each there are, and this is only a 2-state system, then you can use your old friend, the Boltzmann relationship, to calculate the Gibbs free energy difference between the conformations. And so you can do that, and now suddenly you have the relative Gibbs free energy of the 2 states, but also the Gibbs free energy as a function of psi, because you're operating at room temperature and there's some thermal energy. And so here you have now the Gibbs free energy landscape for example, polyl-lysine and polyglutamic acid. And for polyl-lysine, what you see is the minimum energy conformation is PP2. The 2.51 helix is 74 calories per mole. Higher in energy, you can plot out the shape of the potential function. So you learn a lot of information. For polyglutamic acid, you have a shorter side chain, and the minimum is the 2.51 helix. Now, this is old news. This is stuff that we discovered 5 to 10 years ago. What I'm now going to tell you about is something I'm talking about for the first time, and that is looking at the energy landscape in order to find mechanistic information about fibrillization. And so what we're going to do is we're going to take a look at the different conformations that can exist and try to understand how you can go from monomer solution conformation to aggregates, to fibrils. And so that's presently what we're working on. I want to make sure that I cite all of the recent wonderful work by Igor Levna's group on fibrillization. A lot of the work I'm going to tell you now is inspired by their work and their collaborative work with Larry and Rena. And so the story is, as Larry briefly mentioned, is that many proteins will aggregate and fibrilize. And it seems like all proteins will do this under appropriate conditions, and this looks very irreversible for most systems. This results in numerous diseases that can be devastating in consequence. And what you would like to understand is what is the mechanism by which this fibrillization occurs. Now, we're looking into one case, which is a polyglutamine fibrillization. This is a disease that causes Huntington's. And in the case of Huntington's, what you'll see is that there are two possible aggregate states that are stabilized between two types of hydrogen bonding. For most proteins that aggregate, you have hydrogen bonding of the peptide backbone, and that gives you a beta state-like structure. But in polyglutamine, you have an additional side chain that is happy to hydrogen bond. And this complicates the issue. And there's a specific type of fibril that forms, maybe more than one fibril that forms. And the question is, through what mechanism does this occur? And so I'm just showing you an overview of the possible mechanisms. And I'm telling you ahead of time what we've recently discovered in one set of poly-Q peptides. That we have a set of peptides that we have discovered is in a polyproline 2, 2.51 equilibrium, like the polyl-lysine I told you, that was previously thought to be random coil. We can also place the system in a beta hairpin. And the question is, how does it fibrilize to form the final fibril? And what is the conformation of that fibril for this poly-Q? So the idea is we make the fibril, we study the monomer solution, we try to capture the aggregate solution, we study the fibrils, and hope to get insight into the structure and the mechanism. Let me remind you again that we're talking about a poly-Q, a glutamine. So we have the peptide bond, where you have a peptide formed from many Qs. The side chain is a primary amide, and it can do its hydrogen bonding. We found a very unusual poly-Q, which has 2 aspartic acids and 2 lysines at either end, and there's 10 glutamines between them. And so this is a peptide where the flanking residues are oppositely charged. And so you can imagine that they might want to fold. Turns out that we can make 2 different solutions of this peptide which don't look so different in polarized microscopy, but whose X-ray diffraction is completely different and whose Raman spectra are completely different in both solution and in the solid. Here's a remarkable set of spectra. These are 4 spectra, 2 from form 1 and 2 from form 2. These 2 spectra that look very similar to each other look like a little bit like beta sheet, have some turns, and most resemble beta hairpin. So we make a solution of this, and then we evaporate the water, and we have a solid of this. And the solution and solid spectra are very reminiscent, and they go back to one another all day long. On the other hand, we can make a different type of solution where the Raman spectra are completely different in solution. It looks like an extended structure. And it's, in fact, a 2.51 helix pp2 equilibrium. And both the solution and solid state have similar conformations. And these are interconvertible. They're not interconvertible between them unless you heat them. They're stable like for a week. Although, if you abuse the beta hairpin, you can get it to fibrilize. If you start getting it to fibrilize at the early stage, you can take a little bit of it and put it in the pp2 solution, 2.51, and you will initiate fibrillization. So those are just facts. So this just restates what I said. Form 1 is in a pp2 and 2.51 helix, while form 2 is a beta hairpin, and it's a comparison of the solution spectra. Now what you can see here is the amide 3 bands are quite different because the psi angles are different between these conformations. You don't see much of a change in the amide 1 frequency of the peptide bond or the side chain. And it turns out that if we calculate now the psi angle distributions using the methodology I described previously, you find that form 1 is this pp2 2.51 helix conformation and equilibrium, while the other is the beta sheet, and that you can see that you have turns as well that enable the beta hairpin conformation, which we propose looks like this compared to the extended conformation. We can also study the hydrogen bonding of the side chain. Turns out the pi to pi star transition of the side chain amide is redshifted compared to the backbone. And so if you take spectra at 198 nanometers, your spectrum is enriched with the amides of the side chain versus 204, where it's enriched in the amide of the backbone. And by doing the different spectra, you can pick out the different amide 1 vibrations, which are highly diagnostic of the hydrogen bonding. And in studies of valeramide, for example, which is an alkyl substituted amide group, you can compare what the resonance Raman spectra is of that primary amide in water versus acetonitrile, and from the relative intensities and from the frequencies determine the hydrogen bonding. And what we discover is that for this poly-Q peptide, everything is hydrogen bonded to water. There is no interpeptide hydrogen bonding. And so that's what you find for both the beta hairpin and the extended conformation. We've been trying to understand these conformations in a collaboration with Jeff Madura's calculational group at Duquesne. And here's a comparison of our experimental psi distribution to what he calculates now with his updated force fields. And you see there's very good agreement. Once the calculational people know the answer, they can arrive at it instantaneously. Shh. Okay, so if you abuse the beta hairpin samples, then they will fibrilize. And what you see, if you do powder diffraction from these fibrils, is the patterns are very similar. The spacings are very similar. And you can't really determine what the conformation is from the X-ray. Here are these two fibril structures. You can look at them all day and say they look a little different, but not very different. You can compare the fibril Raman spectra to that of the solution, and you can easily see that the solution spectra are characteristically broader than the fibrils, but the two fibrils are almost identical, although the amide 3.3 points out that the spectra are a little different. We're in the midst of probing this. We have not figured all this out yet, but there are differences that we can interpret. We can also do some other experiments. For example, we can form our fibrils in D2O, and then take our fibrils and put them in H2O. Igor Lednev has done some of these preliminary experiments. And what happens then is that those portions of the fibril that are exposed can exchange. And so the peptide bonds and the amides that exchange end up showing very different spectra. From those spectral differences, you can get insight into the conformations. And so we have fibrils we've grown at high concentration where we have side distributions that are quite different from the outside that is growing than from the inside that's the core. And so, for example, these are peptides grown from the PP2 2.51 conformations, seeded by the beta sheet fibrils. And you see the core, which is one color, and the outside, which is another color, have quite different distributions. Whereas if you grow the fibril out of, I'm sorry, I said it wrong. This is the beta hairpin conformation. Whereas if you do it from the extended conformations, both the core and the outside have the same side distributions. And so this addresses the question, what is the mechanism of fibrillization? What you have is you have a sample which has a minimum for the PP2 conformation. You also, in that same sample, have an energy minimum for the 2.51 helix. And the activation barrier is not very large because you're in an equilibrium and they are exchanging between the conformations. On the other hand, there's this gigantic energy barrier between the extended conformations and the beta hairpin conformation. It also has its minimum, and what we know is that you can have fibrillization much faster through the beta hairpin to, well, we don't know what else. That's the whole question of the study. And it looks right now that you have to traverse this form of the reaction coordinate with the PP2 and the 2.51 to get into a beta hairpin, then to eventually the fibril. And it looks like most of the action is at the backbone, not in the side chains in this question. So it's getting late, and thank you for your attention, and I'm pleased to answer questions. And these days in the United States, it looks like we're working this backwards.
No questions? Oh, Professor Nafi.
Do you know anything about the infrared frequency of the 2.5 helix? I know PP2 and beta sheet, but I'm just not sure whether we see any evidence for the 2.5.
Okay, I mean, the situation is the most diagnostic band for these conformations, the amide 3. And this band only occurs in the protonated form. So it's really challenging to try to measure the amide 3 in the infrared. But you can't do the radiant. So the answer is no.
But it's fairly close in the Ramachandran plot.
The 2.51 is like 170, and the PP2 is 145.
There's one more question.
I'd like to go back to the myoglobin. Can you see any activity with the heme group itself and see any changes with binding of oxygen?
Oh, there are characteristic changes of the heme in-plane ring modes, the very large changes in the electronic absorption spectrum. And so there's been an infinite amount of work on hemoglobin, myoglobin, and those kinds of systems, and the correlation of the resonance Raman spectrum and ligand binding back in the 80s and 90s.
Okay. Well, thank you very much, Professor Asher.
Let me give you this.